Hello everyone, welcome to BeBiological. I am your instructor Aditi Bhattacharyya and today we will discuss about cell, the fundamental unit of life. Now what is cell? Cell is the basic structural and functional unit of living organisms. In other words, cells make up living things and carry out activities that keep a living thing alive. Now, what is cell theory? Cell theory is a collection of ideas and conclusions from many different scientists over time that describes cells and how cells operate. Three main features of cell theory are: all known living things are made up of one or more cells. All living cells arise from pre-existing cells by division. The cell is the basic unit of structure and function in all living organisms. Now, this is the timeline of cell theory. These are the major events of discovery of cell and cell theory. First one is the discovery of cell itself by Robert Hooke in 1665. Then observing of living cell by Anton van Leeuwenhoek took place in 1674. Then discovery of nucleus took place by Robert Brown in 1883. Between 1674 and 1883 there are other major events also. Let's look at them. Felix Dujardin in 1835 discovered fluid content of the cell. Then in 1838, Sladen proposed all plants are made up of cells. This is very important of cell theory. Next in 1839, Parkinson named fluid content of cell as protoplasm. Then in 1839, Theodore Swan proposed all animals are made up of cells. This is another important theory of cell. In 1845, Brown proposed cell is the basic unit of life. And then in 1855, Virchow proposed all cells arise from pre-existing cells. Organisms are of two kinds according to the presence of cells. First one is unicellular organisms, which is made up of only one cell, such as Euglena, Paramecium, and yeast. An organism that is made up of more than one cell is called as multicellular organisms like plants, animals, fungus, etc. Multicellular organisms look somewhat like this under the microscope. First picture is of a leaf cell, second one is of muscle cells. The diversity of cells are really high according to the size and the shape of the cells. Most cells are very small or microscopic, some may be very large or macroscopic. The unit used to measure size of the cell is micrometer. The smallest known cell is Mycoplasma, size is about 0.1 micrometer, and the largest cell known is ostrich egg, which size is about 18 centimeter. The largest known human cell is ovum cell, whose size is around 120 micrometer. The smallest known cell is sperm cell, whose size is around 5 micrometer, while the longest known cell in human is nerve cell, whose size is around 1 meter, though not all nerve cells are around this size. Cells varies in shape also. Variation depends mainly upon the function of cells. Some cells like Euglena and amoeba can change their shape, but most cells have a fixed shape. Human RBC or red blood cells are circular, biconcave for easy passage through human capillaries. Nerve cells are branched to conduct impulse from one point to another. Human WBC or white blood cells can change their shape to engulf the microorganisms that enter the body. The detailed structure of a cell has been studied under compound microscope and electron microscope. Certain structures can be seen only under an electron microscope. The structure of a cell as seen under an electron microscope is called ultrastructure. Now this is an animal cell which is composed of many different organelles and compounds such as nucleus, Golgi body, vesicle, plasma membrane, mitochondria, cytoskeleton, centriole, lysosome, cytoplasm and endoplasmic reticulum. A plant cell has also various components like nucleus, Golgi body, vesicle, lysosome, plasma membrane, mitochondria, chloroplast, cell wall, vacuole, endoplasmic reticulum, etc. Chloroplast and cell wall can only be seen in plant cell but not in animal cell. In case of a bacterial cell, capsule, cell wall, plasma membrane, cytoplasm, flagella, food granule, plasmid DNA, ribosomes, nucleoid and pili can be observed. If we study a cell under a microscope, we would come across three features in almost every cell: plasma membrane, nucleus, cytoplasm. Under cytoplasm, we can see cytosol and cell organelles, while under cell organelles, we can see endoplasmic reticulum, Golgi body, lysosome, vacuoles, mitochondria, plastids, centrosome, and cytoskeletons. All activities inside the cell and interactions of the cell with its environment are possible due to these features. Plasma membrane are extremely delicate, thin, elastic, living and semi-permeable membrane made up of two layers of lipid molecules in which protein molecules are floating. Thickness varies from 75 to 110 angstrom. Can be observed under an electron microscope only. The main functions of plasma membrane are: they maintain shape and size of the cell, they protect internal contents of the cell, it regulates entry and exit of substances in and out of the cells, and maintain homeostasis. A cell wall, which can only be seen in plant and bacterial cell, a non-living and outermost covering of the cell. They can be tough, rigid and sometimes flexible, made up of cellulose, hemicellulose and pectin. May be thin or thick multi-layered structure. Thickness varies from 50 to 100,000 angstroms. The functions of cell walls are: provides definite shape, strength and rigidity to the cell. They prevents drying up or desiccation of cells. They helps in controlling cell expansion. It protects cell from external pathogens. Nucleus are the dense spherical body located near the center of the cell. Their diameter varies from 10 to 25 micrometer. They present in all the cells except mature red blood cells in case of animals and sieve tube cells in case of plants. Well developed in plant and animal cells, but underdeveloped in bacteria and blue-green algae or cyanobacteria. Most of the cells are uninucleated or having only one nucleus. Few types of cells have more than one nucleus, or you can say multinucleate. For example, skeletal muscle cells. Let's see the structure of the nucleus. Nucleus has a double-layered covering called nuclear membrane. Nuclear membrane has pores of diameter about 80 to 100 nanometer. Colorless, dense sap present inside the nucleus known as nucleoplasm. Nucleoplasm contains round-shaped nucleolus and network of chromatin fibers. Fibers are composed of deoxyribonucleic acid or DNA and protein histone. These fibers condense to form chromosomes during cell division. Chromosomes contain stretches of DNA called genes. Genes transfer the hereditary information from one generation to the next. The functions of nucleus are control all the cell activities like metabolism, protein synthesis, growth, and cell division. Nucleolus synthesizes ribonucleic acid or RNA to constitute ribosomes. They store hereditary information in genes. Coming to cytoplasm, which is a jelly-like material formed by 80% of water, present between the plasma membrane and the nucleus, contains a clear liquid protein called cytosol and various particles. Particles are proteins, carbohydrate, nucleic acid, lipids, and inorganic ions. Also contains many organelles with distinct structure and function. Some of these organelles are visible only under an electron microscope, granular and dense in animal cells and thin in plant cells. Endoplasmic reticulum, a network of tubular and vesicular structures which are interconnected with one another. Some parts are connected to the nuclear membrane while others are connected to the cell membrane. Two types of endoplasmic reticulum are there. One is smooth endoplasmic reticulum, which lacks the ribosome, and second is rough endoplasmic reticulum, which is studded with ribosomes. The functions of endoplasmic reticulum are: they gives internal support to the cytoplasm. Rough endoplasmic reticulum or RER synthesize secretory proteins and membrane proteins, while SER or smooth endoplasmic reticulum synthesize lipids for cell membrane. In liver cells, SER detoxify drugs and poisons. In muscle cells, SER store calcium ions. Now the next important cell organelle is Golgi body, which is discovered by Camillo Golgi, formed by stacks of 5 to 8 membranous sacs. Sacs are usually flattened and are called the cisternae. They have two ends, cis face situated near the endoplasmic reticulum and the trans face situated near the cell membrane. Their functions are modifies, shorts, and packs materials synthesized in the cell. They deliver synthesized materials to various targets inside the cell and outside the cell. They produces vacuoles and secretory vesicles. They form plasma membrane and lysosome. Look at the detailed work done by a Golgi body. The synthesized material inside the cell can be secreted outside by the Golgi body. They can be stored inside in the form of vesicles, and they can also form lysosome. Lysosomes are small spherical single membrane sac. They found throughout the cytoplasm. They filled with hydrolytic enzymes, occur in most animal cells and in few type of plant cells. The functions of lysosomes are: they help in digesting of large molecules. They protect the cell by destroying foreign invaders like bacteria and viruses by bursting themselves. That is why they are known as suicidal bag. The degradation of worn out organelles can also be done with the help of lysosome. They perform autolysis in dead cells. Vacuoles. are single membrane sac filled with liquid or sap, means water, sugar and ions. In case of animal cells, vacuoles are temporary, small in size and few in number. In plant cells, vacuoles are large and more in number, may be contractile or non-contractile. The functions of vacuoles are: they can store various substances including waste products, they maintain osmotic pressure of the cell, they store food particles in amoeba cells, they provide turgidity and rigidity to plant cells. Next important cell organelle is mitochondria, which are small rod-shaped organelles bounded by two membranes. One is inner, another one is outer. Outer membrane is smooth and encloses the contents of mitochondria. Inner membrane is folded in the form of self-like inward projections called cristae. Inner cavity is filled with matrix which contains many enzymes. They contain their own DNA, which are responsible for many enzymatic functions. That is why they are also known as semi-autonomous cell organelle. The function of mitochondria are: they synthesize energy-rich compound or ATP, ATP molecules provide energy for the vital activities of cells. That is why mitochondria are also known as energy house of the cell. Plastids are double membrane bound organelles found inside plants and some algae. They lack in animal cell. They are responsible for activities related to making the storing of food. They often contain different types of pigments that can change the color of the cell. Plastids are of three types: chromoplast, chloroplast and leucoplast, while leucoplast can be divided into three again: elioplast, amyloplast and proteoplast. Chromoplasts are plastids that produce and store pigments. They are responsible for different colors found in leaves, fruit, flowers and vegetables. For example, carotene is the pigment for carrot, xanthophyll is the yellow pigment for mango, lycopene is the pigment for tomato. Leucoplasts are colorless plastids that store foods. They found in storage organs such as fruits, tubers and seeds. For example, starch is stored as food in case of potato tubers. Protein is stored in case of maize grain. Oil is stored in case of castor seeds. Chloroplasts are double membrane bound organelles found mainly in plant cells, usually spherical or discoidal in shape. Shows two distinct regions, grana and stroma. Grana are stacks of thylakoids, membrane bound flattened discs. Thylakoids contain chlorophyll molecules which are responsible for photosynthesis. Stroma is a colorless, dense fluid. The functions of chloroplasts are: convert light energy into chemical energy in the form of food. They provide green color to the leaves, stems and vegetables. Now centrosome, which you can find only in animal cell. Centrosomes is the membrane-bound organelle present near the nucleus. Consists of two structures called centrioles. Centrioles are hollow cylindrical structures made of microtubules. Centrioles are arranged at right angles to each other. Their functions are to form spindle fibers which help in the movement of chromosomes during cell division. They help in the formation of cilia and flagella. Now, cytoskeleton can be formed by microtubules and microfilaments. Microtubules are hollow tubules made up of protein called tubulin. Microfilaments are rod-shaped thin filaments made up of protein called actin. The functions of cytoskeleton is to determine the shape of the cell. It gives structural strength to the cell, and they are responsible for cellular movements. Now, cells are divided into two broad categories. One is prokaryotic cells or primitive nucleated cell, and another one is eukaryotic or new nucleated cells. The difference between these two are prokaryotic cells nucleus is underdeveloped, while nucleus is well developed in case of eukaryotic cell. Only one chromosome is present in case of prokaryotic cell, while more than one chromosomes are present in case of eukaryotic cell. Membrane-bound cell organelles are absent in case of prokaryotic cell, but all the membrane-bound cell organelles like mitochondria, Golgi body can be found in case of eukaryotic cells. The size ranges from 0.5 to 5 micrometer in case of prokaryotic cell, while the size ranges from 5 to 100 micrometer in case of eukaryotic cell, clearly saying that the prokaryotic cells are smaller than eukaryotic cell. For example, bacteria and blue-green algae are prokaryotic cells, while all the other organisms like all the plants, animals are under eukaryotic cells. Now here are some major differences between animal cell and plant cells. Animal cells are generally small in size, while the plant cells are generally large in size. Cell wall is absent in case of animal cell, while cell wall is present in case of plant cells. Plastids are absent in case of animal cells, but plastids like chromoplastid, chloroplastid, leucoplastids can be found in plant cells. The vacuoles are smaller in size and less in number in case of animal cell, while vacuoles are large in size and more in number in case of plant cells. Centrioles are present in animal cells and centrioles are absent in case of plant cell. This is all about cell. I hope you liked this video. If this video helped you, then please consider subscribing to my channel and share this video to your friends too. Thank you for listening to me.